in the intricate latticework of your gut. Each square millimeter harbors over 100 billion microbial cells. These microbes ferment dietary fibers into short-chain fatty acids, or SFAs, at a rate of 3 to 5 grams per day. CFAs are more than mere byproducts. They are the linchpins that maintain your mucosal barrier's integrity. This barrier, a mere 0.1 millimeters thick, is your frontline defense against pathogens and toxins. The mechanism relies on coupled thresholds. SCFA production must align with mucus layer enhancement to prevent IBD onset. If these thresholds misalign, the barrier weakens, exposing you to chronic inflammation and pathogen invasion. In the failure state, the mucus layer thins to a critical threshold, allowing pathogens to breach the barrier and trigger an inflammatory cascade. The cascade can lead to systemic issues far beyond the gut. Understanding how these coupled thresholds interact within the microscopic dance of your gut microbiome reveals the key to maintaining a fortified inner lining. Let's dive deeper into this intricate mechanism and uncover its secrets. Colonoscopy reveals a stark truth. IBD patients show a significant reduction in SCFAs, measured in micromoles per gram of colon tissue. This deficit marks the threshold where mucosal integrity begins to falter. In healthy individuals, SCFAs maintain the gut lining's robust barrier. Below this critical level, the protective shield weakens, allowing pathogens and toxins to breach. The decline in SCFA levels is driven by disruptions in microbial fermentation, often due to diet or antibiotic use. These disruptions lead to a thinner mucus layer, exposing the gut to chronic inflammation. Johnson et al. 2020. Used in vitro cell culture to investigate the impact of SCFAs on mucus production. Their findings revealed a significant increase in mucus when gut cells were exposed to SCFAs. The experiment established that SCFAs push mucus production above a crucial threshold, reinforcing the gut lining's protective barrier. Li et al. 2019 analyzed population statistics using advanced imaging techniques. They found that IBD patients show significant mucosal layer breakdown, measured in micrometers. This data highlights a critical threshold. The mucosal layer must maintain a minimum thickness to prevent inflammatory bowel disease. The cellular processes ensuring this thickness involve the activation of specific genes by CFAs, which upregulate mucus production and enhance barrier integrity. Wang et al. Using mass spectrometry in a 2017 study, found that CFAs bind to mucus glycoproteins at concentrations greater than 5 millimolar. This binding is crucial for enhancing the stability and integrity of the mucosal layer. At these concentrations, CFA, like molecular anchors, reinforcing the mucus barrier against physical and chemical stressors. The threshold of 5 mm is a critical benchmark. Below this level, the mucus layer becomes more susceptible to degradation and pathogen penetration. Patel et al. 2021. Flow cytometry revealed that immune cell receptor activation increases significantly in the presence of SCFAs. The experiment reveals a critical threshold. CFAs must reach a specific concentration to activate these receptors effectively. Below this level, immune cells remain dormant, failing to mount an adequate defense. Above it, the mucosal barrier is fortified, maintaining its integrity against pathogens and inflammation. The precision of this mechanism underscores the delicate balance required for optimal gut health. Chen et al. 2016. Cow in vivo mouse model. The study found a significant reduction in chronic inflammation when SCFAs were present. Within the gut, adequate SCFAs prevent the breakdown of the mucosal lining. Without them, the barrier weakens, allowing pathogens to breach and trigger chronic inflammation. This elegant mechanism underscores the precision required for optimal gut health. The threshold set by this study defines the minimum level of SCFAs needed to maintain the integrity of the mucosal barrier. When these thresholds are disrupted, the consequences can be severe. A thinning mucus layer and weakened immune response create a perfect storm for pathogen invasion. This breach can lead to systemic inflammation, affecting not just the gut but also distant organs like the liver and brain. Chronic inflammation can trigger a cascade of biological responses, leading to tissue damage, organ dysfunction, and systemic diseases such as metabolic syndrome and cardiovascular disease may follow. The interplay between these thresholds is crucial for maintaining overall health and preventing systemic breakdowns. The alignment of these 6 thresholds, from SCFA production to chronic inflammation prevention, underscores the intricate mechanisms required for a healthy gut. Each threshold acts as a checkpoint, ensuring optimal parameters. Consider the first step. Fermentation in the colon produces CFAs, primarily acetate, propionate, and butyrate. These compounds are not just metabolic byproducts, they are signaling molecules that communicate with the cells of the mucosal lining. When CFA bind to specific receptors on these cells, they trigger a cascade of biological processes. This cascade includes the upregulation of genes responsible for tight junction proteins. These proteins form physical barriers between cells. CFA help modulate immune cell activity, such as T regulatory cells and macrophages, preventing excessive inflammation that can damage the mucosal lining and compromise its integrity. The third threshold is the maintenance of the mucus layer. SCFA stimulate the production of mucus, a gel-like substance that coats the gut lining and provides an additional barrier against harmful microorganisms. This mucus layer is dynamic and responsive to changes in SCFA levels, ensuring it remains effective in its protective role. Without sufficient CFAs, the mucus layer thins, making the gut more vulnerable to infection. The fourth threshold involves the regulation of gut motility. CFA influence the rate at which food moves through the digestive tract. Proper motility ensures that nutrients are absorbed efficiently and waste is eliminated effectively. This balance is critical for overall gut health, as disruptions can lead to conditions like irritable bowel syndrome. The fifth threshold involves the regulation of blood flow. CFAs stimulate the dilation of blood vessels in the gut, increasing blood flow and nutrient delivery to the mucosal lining. Adequate blood flow ensures that these cells receive the necessary nutrients and oxygen to maintain their integrity. The final threshold is the prevention of chronic inflammation. A significant reduction in chronic inflammation when SCFAs are present highlights their critical role. This reduction underscores the importance of maintaining adequate levels of SCFAs to prevent the breakdown of the mucosal barrier and subsequent inflammation. The interplay between these thresholds is a testament to the elegance and precision of biological systems. Each step in this process is finely tuned, ensuring that the gut's mucosal barrier remains strong and effective. As we reflect on the intricate mechanisms that keep our gut healthy, what does this reveal about the broader systems in our body? In the next episode, we'll explore how these same principles apply to the brain's blood barrier and its role in cognitive health.